The Y chromosome is one of two sex chromosomes in mammals, including humans, and many other animals. The other is the X chromosome. Y is the sex-determining chromosome in many species, since it is the presence or absence of Y that determines the male or female sex of offspring produced in sexual reproduction. In mammals, the Y chromosome contains the gene SRY, which triggers testis development. The DNA in the human Y chromosome is composed of about 59 million base pairs. The Y chromosome is passed only from father to son, with a 30% difference between humans and chimpanzees. The Y chromosome is one of the fastest evolving parts of the human genome. To date, over 200 Y-linked genes have been identified. All Y-linked genes are expressed in hemizygous except in the cases of aneuploidy such as XYY syndrome or XXYY syndrome. Overview. Discovery. The Y chromosome was identified as a sex-determining chromosome by Nettie Stevens at Bryn Mawr College in 1905 during a study of the mealworm Tenebrio mollitor. Edmund Beecher Wilson independently discovered the same mechanisms the same year. Stevens proposed that chromosomes always existed in pairs and that the Y chromosome was the pair of the X chromosome discovered in 1890 by Hermann Henking. She realized that the previous idea of Clarence Irwin McClung, that the X chromosome determines sex, was wrong and that sex determination is, in fact, due to the presence or absence of the Y chromosome. Stevens named the chromosome Y, simply to follow on from Henking's X alphabetically. The idea that the Y chromosome was named after its similarity in appearance to the letter Y is mistaken. All chromosomes normally appear as an amorphous blob under the microscope and only take on a well-defined shape during mitosis. This shape is vaguely X-shaped for all chromosomes. It is entirely coincidental that the Y chromosome, during mitosis, has two very short branches which can look merged under the microscope and appear as the descender of a Y shape. Variations. Most mammals have only one pair of sex chromosomes in each cell. Males have one Y chromosome and one X chromosome, while females have two X chromosomes. In mammals, the Y chromosome contains a gene, SRY, which triggers embryonic development as a male. The Y chromosomes of humans and other mammals also contain other genes needed for normal sperm production. There are exceptions, however. For example, the platypus relies on an XY sex determination system based on 5 pairs of chromosomes. Platypus sex chromosomes in fact appear to bear a much stronger homology with the avian Z chromosome, and the SRY gene, so central to sex determination in most other mammals, is apparently not involved in platypus sex determination. Among humans, some men have 2 X's and a Y, or 1 X and 2 Ys, and some women have 3 X's or a single X instead of a double X. There are other exceptions in which SRY is damaged, or copied to the X. For related phenomena, see androgen insensitivity syndrome and intersex origins and evolution. Before Y chromosome, many ectothermic vertebrates have no sex chromosomes. If they have different sexes, sex is determined environmentally rather than genetically. For some of them, especially reptiles, sex depends on the incubation temperature. Others are hermaphroditic. Origin. The X and Y chromosomes are thought to have evolved from a pair of identical chromosomes, termed autosomes. When an ancestral mammal developed an allelic variation, a so-called sex locus simply possessing this allele caused the organism to be male. The chromosome with this allele became the Y chromosome, while the other member of the pair became the X chromosome. Over time, genes which were beneficial for males and harmful to females either developed on the Y chromosome, or were acquired through the process of translocation. Until recently, the X and Y chromosomes were thought to have diverged around 300 million years ago. However, research published in 2010, and particularly research published in 2008 documenting the sequencing of the platypus genome, has suggested that the XY sex determination system would not have been present more than 166 million years ago, at the split of the monotremes from other mammals. This re-estimation of the age of the variant XY system is based on the finding that sequences that are on the X chromosomes of marsupials and eutherian mammals are present on the autosomes of platypus and birds. The older estimate was based on erroneous reports that the platypus X chromosomes contained these sequences. Recombination inhibition. Recombination between the X and Y chromosomes proved harmful. It resulted in males without necessary genes formerly found on the Y chromosome, and females with unnecessary or even harmful genes previously only found on the Y chromosome. As a result, genes beneficial to males accumulated near the sex-determining genes, and recombination in this region was suppressed in order to preserve this male-specific region. Over time, the Y chromosome changed in such a way as to inhibit the areas around the sex-determining genes from recombining at all with the X chromosome. As a result of this process, 95% of the human Y chromosome is unable to recombine. Only the tips of the Y and X chromosomes recombine. The tips of the Y chromosome that could recombine with the X chromosome are referred to as the pseudo-autosomal region. The rest of the Y chromosome is passed on to the next generation intact. It is because of this disregard for the rules that the Y chromosome is such a superb tool for investigating recent human evolution from a male perspective. Degeneration. By one estimate, the human Y chromosome has lost 1,393 of its 1,438 original genes over the course of its existence, and linear extrapolation of this 1,393 gene loss over 300 million years gives a rate of genetic loss of 4.6 genes per million years. Continued loss of genes at the 4.6 genes per million year rate would result in a Y chromosome with no functional genes, that is, the Y chromosome would lose complete function within the next 10 million years, or half that time with the current age estimate of 160 million years. Comparative genomic analysis reveals that many mammalian species are experiencing a similar loss of function in their heterozygous sex chromosome. Degeneration may simply be the fate of all non-recombining sex chromosomes, due to three common evolutionary forces: high mutation rate, inefficient selection, and genetic drift. However, comparisons of the human and chimpanzee Y chromosomes show that the human Y chromosome has not lost any genes since the divergence of humans and chimpanzees between 6 to 7 million years ago, and a scientific report in 2012 stated that only one gene had been lost since humans diverged from the rhesus macaque 25 million years ago. These facts provide direct evidence that the linear extrapolation model is flawed and suggest that the current human Y chromosome is either no longer shrinking or is shrinking at a much slower rate than the 4.6 genes per million years estimated by the linear extrapolation model. High mutation rates. The human Y chromosome is particularly exposed to high mutation rates due to the environment in which it is housed. The Y chromosome is passed exclusively through sperm, which undergo multiple cell divisions during gametogenesis. Each cellular division provides further opportunity to accumulate base pair mutations. Additionally, sperm are stored in the highly oxidative environment of the testis, which encourages further mutation. These two conditions combined put the Y chromosome at a greater risk of mutation than the rest of the genome. The increased mutation risk for the Y chromosome is reported by Graves as a factor 4.8. However, her original reference obtains this number for the relative mutation rates in male and female germlines for the lineage leading to humans. inefficient selection. Without the ability to recombine during meiosis, the Y chromosome is unable to expose individual alleles to natural selection. Deleterious alleles are allowed to hitchhike with beneficial neighbors, thus propagating maladapted alleles into the next generation. Conversely, advantageous alleles may be selected against if they are surrounded by harmful alleles. Due to this inability to sort through its gene content, the Y chromosome is particularly prone to the accumulation of junk DNA. Massive accumulations of retrotransposable elements are scattered throughout the Y. The random insertion of DNA segments often disrupts encoded gene sequences and renders them non-functional. However, the Y chromosome has no way of weeding out these jumping genes. Without the ability to isolate alleles, selection cannot effectively act upon them. A clear, quantitative indication of this inefficiency is the entropy rate of the Y chromosome. Whereas all other chromosomes in the human genome have entropy rates of 1.5–1.9 bits per nucleotide, the Y chromosome's entropy rate is only 0.84. This means the Y chromosome has a much lower information content relative to its overall length. It is more redundant. Genetic drift. Even if a well-adapted Y chromosome manages to maintain genetic activity by avoiding mutation accumulation, there is no guarantee it will be passed down to the next generation. The population size of the Y chromosome is inherently limited to 1/4 that of autosomes. Diploid organisms contain 2 copies of autosomal chromosomes, while only half the population contains one Y chromosome. Thus, genetic drift is an exceptionally strong force acting upon the Y chromosome. Through sheer random assortment, an adult male may never pass on his Y chromosome if he only has female offspring. Thus, although a male may have a well-adapted Y chromosome free of excessive mutation, it may never make it into the next gene pool. The repeat random loss of well-adapted Y chromosomes, coupled with the tendency of the Y chromosome to evolve to have more deleterious mutations rather than less for reasons described above, contributes to the species-wide degeneration of Y chromosomes through Muller's ratchet. Gene conversion. As it has been already mentioned, the Y chromosome is unable to recombine during meiosis like the other human chromosomes. However, in 2003, researchers from MIT discovered a process which may slow down the process of degradation. They found that human Y chromosome is able to recombine with itself using palindrome-based bare sequences. Such a recombination is called gene conversion. In the case of the Y chromosomes, the palindromes are not non-coding DNA. These strings of bases contain functioning genes important for male fertility. Most of the sequence pairs are greater than 99.97% identical. The extensive use of gene conversion may play a role in the ability of the Y chromosome to edit out genetic mistakes and maintain the integrity of the relatively few genes it carries. In other words, since the Y chromosome is single, it has duplicates of its genes on itself instead of having a second, homologous, chromosome. When errors occur, it can use other parts of itself as a template to correct them. Findings were confirmed by comparing similar regions of the Y chromosome in humans to the Y chromosomes of chimpanzees, bonobos and gorillas. The comparison demonstrated that the same phenomenon of gene conversion appeared to be at work more than 5 million years ago, when humans and the non-human primates diverged from each other. Future evolution. In the terminal stages of the degeneration of the Y chromosome, other chromosomes increasingly take over genes and functions formerly associated with it. Finally, the Y chromosome disappears entirely, and a new sex-determining system arises. Several species of rodent in the sister families Muridae and Chrysetidae have reached these stages, in the following ways: the Transcaucasian mole vole, Elobius lutetiens, the Zazen mole vole, Elobius tancra, and the Japanese spinous country rats Tokudia osimensis and Tokudia tokunoshimensis, have lost the Y chromosome and SRY entirely. Tokudira spp. Have relocated some other genes ancestrally present on the Y chromosome to the X chromosome. Both genders of Tokudira spp. and Elobius lutetiens have an XO genotype, whereas all Elobius tanquae possess an XX genotype. The new sex-determining system for these rodents remains unclear. The wood lemming Myopus schisticolor, the Arctic lemming, Dicrostonyx torquatus, and multiple species in the grass mouse genus Achodon have evolved fertile females who possess the genotype generally coding for males, XY, in addition to the ancestral XX female, through a variety of modifications to the X and Y chromosomes. In the creeping vole, Microtus oregoni, the females, with just one X chromosome each, produce X gametes only, and the males, XY, produce Y gametes, or gametes devoid of any sex chromosome, through non-disjunction. Outside of the rodent family, the black muntjac, Munchicus crinifrons, evolved new X and Y chromosomes through fusions of the ancestral sex chromosomes and autosomes. 1-to-1 sex ratio Fisher's principle outlines why almost all species using sexual reproduction have a sex ratio of 1-to-1, meaning that 50% of offspring will receive a Y chromosome, and 50% will not. W.D. Hamilton gave the following basic explanation in his 1967 paper on extraordinary sex ratios. Given the condition that males and females cost equal amounts to produce, suppose male births are less common than female. A newborn male then has better mating prospects than a newborn female, and therefore can expect to have more offspring. Therefore, parents genetically disposed to produce males tend to have more than average numbers of grandchildren born to them. Therefore, the genes for male-producing tendencies spread, and male births become more common. As the 1-to-1 sex ratio is approached, the advantage associated with producing males dies away. The same reasoning holds if females are substituted for males throughout. Therefore 1-to-1 is the equilibrium ratio.